Um, Hi, Dr. Finley. Your background is in chemical engineering. So, during your early career, what inspired you to shift towards biomedical engineering and cancer research?
Yes, so my undergraduate degree as well as my PhD are both in chemical engineering. And in that context, I studied a lot of chemical reactions and also looked at the dynamics of chemical reactions. So, how fast are the reactions happening, and how fast are the species that are included in the reactions, how fast are their levels changing? So I got, really excited about building, really even as an undergraduate student, building models of chemical reactions. But as I went into my PhD, I wanted to apply that knowledge in the context of human health and disease. So in my PhD, I also worked on looking at metabolic reactions, but mainly to see how we might predict new reactions that could occur to break down toxic compounds. So getting a little bit closer to human health and the relevance of that research. And then I decided to do a postdoctoral fellowship. And it's then in that transition that I said, well, let me look specifically for opportunities that are focused on health and disease. And I stumbled across a fantastic research group at Johns Hopkins that was looking at using mathematical modeling to study tumor angiogenesis, which is the formation of new blood vessels inside. And so I learned that the same kinds of models that I was using to study chemical reactions could be applied to study biochemical reactions and reactions that are happening in biology. So that's really where the transition from chemical engineering to more biomedical engineering occurred.
So, what were some of the more challenging aspects of moving from engineering to cancer research?
Yes, I think the biggest difficulty in moving from chemical engineering to cancer research is just not knowing the biology, right? Cancer is such a complex and heterogeneous disease. Even within one type of cancer, every patient's tumor is very different. So, first of all, just trying to grasp the complexity of the disease, and then also learn about the proteins and the signaling species and the transcription factors that are important in driving tumor growth and also influencing the response to treatment. So I think the biggest hurdle is just comprehending the complexity and learning about the biology.
So, in my research with Dr. Rockney, he emphasized that it's really important to take the most important elements of a model and then sort of simplify the rest because otherwise there's too many factors to
Yes.
keep track of. So how would you in your research, how do you simplify very complex models to something that accurately reflects how cancers respond to treatments?
Yes, I think the approach that we take to building a model that is predictive but still manageable and not too complex is by starting with the biological question. So, really, the question should drive what components you add to the model and also how complex the model is. So you should really only be developing a model that is complex enough to answer the biological question that you set out. So it's really important to have that question at the top of your mind so that it informs your decisions about how to develop the mathematical model. So, as an example, right, if you're interested in a particular signaling pathway and how that pathway influences proliferation of cancer cells, then maybe you don't look at the 20 other pathways that are also influencing cancer cells, but maybe you say, I'm going to focus on this one, but I'm going to look at how that signaling pathway might behave differently depending on the conditions in which the tumor is growing. So it's still capturing what's happening, but in a way that is specific to the question that you're interested in answering.
So I'd like to ask about a specific factor in cancer treatment. Cancer-associated fibroblast in colon cancer. Could you speak a bit about how those impact the treatment as well as the model?
Right. Yes. So one project in the lab is looking at colorectal cancer. We know that this is an important type of cancer to study. It's the second leading cause of cancer-related deaths in men. And the 5-year survival rate is pretty low, maybe just around 10%. And colorectal cancer patients often develop resistance to standard therapies. So there's a lot of motivation for studying colorectal cancer. Now, this is a type of tumor that has a large stromal compartment. So tumors are not just made up of cancer cells. They're made up of a lot of other different kinds of cells. And the stromal compartment is like a combination of multiple different cell types that also influence tumor growth. And one of those cell types is called cancer-associated fibroblasts. So they influence the structure of the tumor. They influence how the cancer cells might respond to treatment. Cancer-associated fibroblasts also take up nutrients, so they're competing with cancer cells and other cells in the tumor microenvironment for the nutrients and the oxygen that are available for growth. So we have focused on colorectal cancer cells and their interactions with cancer-associated fibroblasts because there's some evidence in the literature and also in clinical observations that cancer-associated fibroblasts interact with colorectal cancer cells and maybe even outcompete for nutrients, but some kind of metabolic interaction. So we are focused on that kind of interaction between those two different cell types.
Does this relate to angiogenesis and how yes.
Yeah, not directly. So fibroblasts don't directly influence angiogenesis. So yeah, I don't think there's a strong connection there.
Okay, thank you. So how do um when my research, how do CAR Ts help fight cancer? And I guess there's like a lot of novel treatments recently, like extracellular vesicles, oncolytic viruses, CAR T cells. And how can computational models be used to improve the effectiveness of newer therapies?
Yeah, I think there's a lot of excitement about CAR engineered cells, whether it's T cells or other engineered cells that are now able to express CARs. Because this is immunotherapy, it's using the body's own immune system to target and kill cancer cells. And so I think there are a lot of ways in which modeling can help the field make better progress in this area. I think one way is for us to just understand the dynamics, right? What's the time sequence of events of the cancer cell being recognized by a T cell that's expressing the CAR, and how fast does that recognition happen? How fast does the activation of the CAR T cell happen? And then how does the CAR T cell's activation influence the cancer cell? So how fast does that killing occur? I think the dynamics are very useful and amenable to mathematical modeling. I think another place where models can help is understanding the effect of heterogeneity, right? So every time that a CAR T cell encounters a cancer cell, it's not going to happen with 100% certainty that the T cell will kill the cancer cell, right? There's a probability of that killing occurring. And maybe that probability depends on what a lot of other factors that are happening and present in the tumor. So mathematical modeling can help us understand, well, what happens if that probability of killing goes down to 40% instead of 100%? How many T cells would you need in order to kill the cancer cells that are there? So those kinds of questions where there's a lot of complexity or where there's questions about the effect of variability, I think are places where mathematical modeling could be useful. Another place where modeling can be used in studying CAR T cells is just what's the distribution of the T cells in the body, right? You administer the population of CAR T cells. They're not all, if let's say that 1 million cells are given to the patient, not all 1 million are going to go directly to the tumor, right? They're going to be distributed in different tissues in the body. Most of them will probably go to the liver, which is the way in which a lot of molecules and cells are removed from the body. So then the question is, well, what is that distribution? How fast does it occur? Like, how fast are they moving through the body? And what's the effect of having only 10% of the CAR T cells go to the tumor versus 100%? So those kinds of questions where like there's a uncertainty about the rate at which something happens or the probability with which something happens, those are places where I think modeling can be useful to at least say, well, here's a prediction, a set of predictions about what the effect of that uncertainty might be.
So could you speak a bit about how the importance of, I guess, having each thing as a probability instead of a like so as an average, let's say 40%, 10% will go to the site versus like modeling each individual one as separate path.
Yeah, I think it really then when you get the prediction, right, instead of saying with all like on average 40% of the cells will reach the tumor, let's say. Now let's say you have a distribution of cells that reach the tumor. So then you're able to better understand the effect of variability. So it's not always going to be 100% certainty that something happens, but there's a probability that an interaction will happen or killing will happen, killing of the cancer cell. So it's useful to at least acknowledge that and then incorporate it into a model so that you can make more sound and rigorous predictions.
So I was wondering about biomarkers in cancer treatment and how computational models help predict which therapies are most likely to work for individuals.
Yes, so CAR T cells, for example, the CAR is trying to recognize a certain antigen that's expressed on tumor cells. So naturally then, a biomarker would be, well, if I could profile a cancer patient's tumor cells, and I know whether they express that antigen or not, then that would be a biomarker to say, well, this patient should receive CAR T cell therapy that's targeting this particular antigen. So if the cancer cells are expressing antigen X, then yes, we should give this CAR T cell that targets and recognizes antigen X. So I think that's a very straightforward way of thinking about biomarkers. Maybe another biomarker would be, well, what's the composition of the tumor? So there are some immune cells that inhibit or block the effect of CAR T cells. They're called T regulatory cells or some other suppressive immune cell. And so if the tumor, like again, profiling a patient's tumor, if we can identify how many of those cells that would block the CAR T cell therapy there are, then we might be able to say whether that patient should receive the therapy or not. So biomarkers are very important for classifying which patients should get which drugs and maybe even predicting whether the drug would be effective or not.
One thing they mentioned in Roberts Academy is that science is becoming more interdisciplinary in recent years. So, how do you bring together engineers, biologists, mathematicians in your research, and why is it so crucial for advancing cancer treatment?
Yeah. Well, again, I think it goes back to the point about the complexity of cancer. And so just having one viewpoint, like just the viewpoint from a cancer biologist, I don't think is going to be sufficient in making progress in really treating cancer patients. Instead, you need someone who looks at the system from a different perspective, right? And that's where the interdisciplinary collaborations come in, because something that I don't understand, or that a way that see a problem is different than a way that a mathematician or a physicist or a cancer biologist looks at a problem. So it's nice to have people coming from multiple viewpoints and multiple areas of training, so that they complement one another and we can make better and faster progress towards improving cancer patient outcomes. So I think it's really important. Now, how is it done? Well, I think it's done by like me giving a presentation about the kind of modeling that I do to an audience of biologists, and they can say, well, maybe you're missing this part of the tumor microenvironment that's really, really important. Or maybe you don't really need to worry about another part because it's not so important. So I think the way to promote interdisciplinary research is by people from different disciplines coming together and giving a talk to an audience that they normally wouldn't present to, so that they can get that kind of feedback that is a different perspective from their own.
Have there been any advances in technology, for example, computing power, data collection, or like, I guess things way out there like AI that have impacted your work in cancer therapy research?
Yeah, so definitely having, for example, single cell technology is very useful. That has happened over the past decade. So being able to say, oh, it's not just a homogeneous set of T cells inside of the tumor or inside of a patient, but there's a distribution of T cells, and some are subtype A, and some are subtype B, and some are subtype C. So having single cell technology has been very useful for the kinds of models that we build. I think another utility that has come along is being able to profile a tumor across space. So not just taking the tumor and again treating it as a homogeneous entity, but looking at how much expression of different antigens or how much of different cell populations are present in different locations within the tumor. So single cell technologies, spatial technologies have been really useful. And then on the computational side, yes, like having high performance computing, even AI. So we don't use it so much in my lab, but for example, being able to learn features of a clinical image with AI and then predict what the response to treatment would be or what the next image might look like, because you have a sequence of images and can make that prediction of what comes next. So I think as more data become available and the tools for AI and machine learning improve, then that can also be, a great technology to push the field forward.
Yeah, with all these new tools, what do you hope to see as the next big breakthrough in cancer treatment?
That's a good question. Um, I think it's a breakthrough that the field has been aiming for a while, and that's, personalized medicine. So being able to treat a particular patient's tumor and not just the average patient's tumor, I think is really useful. That will require single cell profiling. I think that will require spatial omics, transcriptomics, proteomics. In our work, we're also very interested in metabolism. So I think spatial metabolomics will also be really important. And so I think personalized medicine, but in a way that still takes in all these different types of data and makes reliable predictions.
Yeah, so I guess I'm interested in joining mathematical oncology as I go to college and beyond. What advice would you say about getting started in not just mathematical oncology, but science and cancer research in general?
Yeah, I think just asking questions and trying to get experiences in research labs, even if you're not, working on a big project, just sitting in on research presentations, on group meeting presentations, and sitting in on presentations where you don't know very much. That's where the real learning occurs, right? Where you feel a little bit uncomfortable that this is just beyond my understanding. That's where you have to learn more, learn faster. And that's where, the exciting changes could happen in your own understanding. So the suggestion would be just to ask questions and immerse yourself in cancer biology research as much as you can.
And as I go into college and beyond, what majors and areas of study would you recommend to like sort of just get into the ideas of oncology and mathematics?
Yeah. I might be biased, but I do think that like engineering or quantitative biology, computational biology, something quantitative is a great discipline to major in because then you have the mathematical foundation for looking at biological questions through this lens of math. And as I did, and as I've learned and seen in colleagues and trainees also, it's a little bit easier to go from the math and learn about the biology than to start in a very biological training discipline and then try to learn more about the math. So I would probably suggest something quantitative like engineering, even like physics or computational biology as the place to start, and then seeing, well, what cancer am I interested in, or how can I learn more about the biology?
Yeah. Again, thank you so much for speaking with me today. One last thing, if there's one thing you hope that the next generation of scientists will carry forwards in the fight against cancer, what would it be?
Hmm. Maybe I think it's really important to keep the patient in mind. I think that's harder to do when you're doing basic science research, so something that's not immediately translational, but realizing that the model that I'm building is meant to represent a patient and thinking about how that patient, like what environment they live in, what things they're exposed to, what their family history is, or the history of their disease is. I think it's easy to do cancer research and sort of leave the patient out of it, especially maybe in mathematical oncology or something that's not so immediately translatable. And so I think keeping that in mind so that you know what the potential impact could be and how important this research is.
Yeah. Thank you so much. Yeah, your advice today, especially with um, I guess like major especially has been helpful. I'm trying to go into college. I'm applying to college soon, so
Mhm. Yeah,
coming really, yeah. Yeah,
great. Yeah, no, this is my pleasure. I'm happy to talk science and careers anytime.
thank you.